I refresh my VPN. I think it's going. Let me know. Yes, I think we're in. Yes, we're live. We're live, right? Good.
Yeah, I think so.
Okay. So good morning, everyone. Is panning from somewhere in the West relative to where I am now today to India, is spanning longitude, latitude, and time. Thank you to our speakers today, for whom it must be, if I remember well, something like 9 p.m. or so. So welcome to another meeting of FINE. It's my great pleasure today to welcome Vivi Robin, who will be presented. This is going to be a team presentation shared by TT Arvind. And before I introduce them and let them take it from here, let me just remind you that we have weekly meetings. And so very quickly, I want to show you next week, we're going to hear from Jen Moss from Virginia Tech University in the US with a talk that's titled Unpacking Thermal Effects on Social Behavior Through Integrative Approaches. That's going to be next week. Today, we have, like I said, Vivi, Robin, and TT, and their talk is going to be Unraveling the Complexities in the Songs of an Endemic Shola Sky Island Bird. Now, I was doing my homework and learning more about the research, and this is what they mean by islands. So it's fascinating. I am particularly interested in their talk because we're doing research also with things we call forest islands in the South American Chaco. Robin is very interested in studying ecological processes as they relate to ecology, to biogeography, to evolutionary ecology. And what he does, I mean, he looks at those questions in the context of island systems. And so today they will be sharing with us some of the research that, from what he was telling me, really spans over some very variable areas. Robin is an associate professor in the Indian Institute of Science, Education, and Research. He's also a National Geographic Explorer. TT is a PhD student already, as she was telling me, in the stages of analyzing and preparing her dissertation research for publication in collaboration with Vivi Robin and his team. Robin has published extensively, and I think that what they're going to be sharing with us really fits so nicely in the FINE seminar series, among other reasons, because they really are very interdisciplinary, and they're really tackling behavioral ecology questions, social evolution questions, with, as he describes in his webpage, with a very explicit, clear desire to connect it to conservation efforts. So many of us sometimes talk about that, but I feel personally that maybe I haven't done enough of concrete, real, on-the-ground conservation efforts. I think that's very, very different from what we're going to hear from Vivi and Chiti. So with that, I think that, Robin, I have already made you a co-host. Like, if we have any new participants, let me remind you, the presentation is going to go roughly 45, 50 minutes. We try to finish before the hour for those who need to go. And after that, we started the question and answer session of FINE, which has really become a signature of the FINE series. We all enjoyed very much staying up after the talk to have a one-on-one discussions and conversations about the research that our colleagues are presenting today. So thank you for joining us at this late in the evening. Robin and Chiti, please take it from here.
Yeah, thank you. I'm Robin, and Chiti, if you want to quickly say hello before we get started, then...
Sure. Hi, everyone. I am Chiti. I'm quite excited for today's talk, and let's start.
Yeah, sure. So, I mean, what I'm going to try and do is set the stage a little bit, starting by describing, the system that we have. That is the landscape that the study is set in. I want to quickly acknowledge that while Chiti and I are talking today, there are two others who are not talking, and their work is also included in the presentation here. That is Rohit Srinivasan and Suyash. I'll explain their roles as we go along. But the landscape that I'm talking about is this. This is the highest mountain in the Western Ghats. This is the kind of landscape that you see. It's a mosaic of forests and grasslands. But before we get there, I just want to quickly explain that our institute is called Indian Institute of Science, Education and Research. It's a mouthful, so we just abbreviate it to IISER. And this one is located near Tirupati. I think you have another speaker from another IISER, which is from Mohali, sometime later on. Our campus is right next to the Eastern Ghats. And this institution itself is a sister institution to IIT, which is the Indian Institute of Technology, one from where you have, I think, the Microsoft and Google CEO and all that coming up. So essentially, we focus on basic education, all sciences, physics, chemistry, math, biology, under the Ministry of Education. It's a fairly new one, set up in 2016. And my group primarily focuses on a few different threads. The first is landscape ecology, because we need to understand. We study birds, but in order to study birds, it turns out that you also need to understand quite a lot about the landscape that they occupy. So that's where the conservation bits that Eduardo was talking about, that's where that comes in. We look at patch sizes, how historically the landscape has changed using remote sensing, satellite imagery data, and so on. But our main bread and butter work is, in these two themes, which is evolution and biogeography, where we go out, collect samples, blood samples from wild birds from across different parts of India, and then bioacoustics and behavior, which is what we're going to talk about. We also, because we are in a unique landscape, we also try and do a little bit of citizen science and outreach to connect with the local communities around here to build capacity in various kinds of biodiversity research. So we have a fairly largest team of about 25 people working across these themes. So a little bit more about the islands. I think one of the first things that strikes people when you talk about islands is that you imagine these oceanic islands, but the islands that what I'm talking about are these high elevation mountain tops. So this is the Western Ghats. This is the southern tip of the peninsular Indian landscape. And along the western coast, you find these various hills, and the top of the mountains is what you see highlighted in these circles. I mean, in this contour. These are all about 1,400 meters. And about 1,400 meters, you find this slightly wet and cold habitats, because we are very coastal, tropical. So you have these montane forests. And what's really interesting is that these mountains, they're isolated from each other, but within each mountain, you also have this matrix of forests and grassland. So you have this very interesting mosaic, of landscape. And of course, one of the other interesting thing for us is that it's a very linear mountain. So it's not like those islands where you have various possibilities of connections. If you have 5 islands, all possibilities are there. Here, because it's linear, a lot of the connections are A to B to C to D and so on. So it's a nice system to have, where we've been exploring quite a lot of, looking at gene flow and phylogenetics and so on. And that is what actually sets the stage for the work that I'm going to describe. Yeah, this is what, if you go into any of these mountain tops, this is what it will look like. It's this mosaic of forests and grasslands. And what are my initial research, especially for my PhD and postdoc actually, where I started collecting blood samples, which were first samples from some of these landscapes. It turned out that some of these species are actually very unique. They were these mountaintops hosted species that were over 5 million years apart. The divergences were quite deep. We didn't realize at that point, but they were actually had to be assigned different genera, because they were very different from all the other groups of species that were there. And one of the species is what we've kind of focused a lot of our work on, This is what we call the Sholikola. That's a scientific name because Shola is the habitat where this bird is found. So we decided to call it Sholikola or the Sholakili, which is the common name now. One of the very interesting things is that the song is very variable. In fact, that is what set me on this journey. Just after my master's, when I was I was trying to do a survey looking for these birds, and I went from one mountain to another, and I noticed that the song is a little bit different on the other mountain from one that I came from. And that's when I started, asking questions with genetics and trying to quantify song. So in this talk, I'm of course going to give you a little bit of the background of what we know about songs of this species, and that's pretty much the only published part that I'm going to talk to you about. I'm going to use this opportunity actually to explore sharing a lot of work that is unpublished and ideas that we are actually exploring. And that's because, like Eduardo said, you have about an hour of interaction, and I would really like to hear, if you have any ideas, because there are some really interesting things that are happening here. So we'll describe how we measured song complexity and syntax for the species. We looked at neighbors, how neighbors share songs. And then Chiti will tell us a little bit about her PhD work, the core of her PhD work, which is looking at population differences in songs and then trying to match that with genetic differences between these populations. And then we'll, quickly talk about female song. This is quite, it's discussed quite a lot, and people are, basically people are finding out that in the tropics, at least, many female passerine birds have songs. And we did not know this until, an intern, undergraduate intern started working with us, and he started quantifying this. Ultimately, I want to kind of bring it back to this big picture, and a lot of it is unsolved. And primarily, we would like to understand a little bit more about why the song of this bird is so complex, and what is actually happening here. This is something that we still don't understand fully. So first thing, I'll play the song of the Sholakili, and of course, this is a spectrogram, and we'll just play the song. You can just follow that along. All right, I'm glad that that came through. We spent hours trying to fix that. Yeah. Anyway, so that's the song from one of the mountains, one of the populations. And early on for my PhD, I looked at songs just across these two mountains. The one with this black star, which is Uti, and then further south. And later I expanded to all of these mountains, and also within the mountain across an east-west gradient. But in the east-west, I only had the extreme samples. And of course, as one can imagine, the songs on all the mountains, all these isolated populations were indeed very different statistically. But what was very interesting was within the same mountain, within the same island, there seemed to be some variation. And yeah, so this is just across all the mountains. What was really interesting, what I found really fascinating, was that the east-west variation seemed to be more in certain parameters of frequency. So across the mountains, it was syntax that was different, but within the mountain, there was this variable which we call complex vocal mechanism, which we think is probably from how the passerines use the dual sirens, both sirens. And we measured this. So how do we know that it's actually dual sirens usage? We can't. All we can say is that it seems to make two sounds at the same time. Most likely it's because of dual sirens usage. But we quantified this. So anything which has less than 2 milliseconds, we count it as a complex vocal mechanism, which is it's making two sounds at the same time. And then we looked at its occurrence across different populations. And it looks like most of the populations can make the sound, these dual syrinx usage, but some populations do it more than some others. So it's basically like saying that there are these cultural traits that just seems to be more common in some population than some others. Yeah. So this is where the published literature on this ends, and like from now on, everything that I'm going to talk to you about is what we are kind of exploring. And yeah, so this is the part that this is the data that I was talking to you about, where the red and the green, these two populations are actually within the same mountain, but they are very different in this complex vocal mechanism. Yeah. So what happened at that point of time was that we had all these results, and we had a really nice field station up on the mountains. And since we have an undergraduate program, one of the interns there walked in, that's Suyash Sawant. He's currently doing a PhD at University of Florida. And he, one thing that he caught on to us about was, what is this complexity that you all talk about? Everybody says that the Sholikula has very complex songs, but have you actually measured this? How do people measure complexity? And then I said, like, figure something out. There must be so many measures of diversity. And he started trying out different measures. And until he said, I'm not really satisfied with many of the measures that are out there. So he comes from a background in physics and math. So he said, I'm going to create a metric of what should be a way to measure a song complexity. And he did that. Basically, what he did was he digitized all of these notes, and he ran this spectral cross correlation, and he created a kind of index called the note variability index, which he then subsequently he improved this. First thing that we said was, how do we know that this is actually measuring something different? Is it because of the data that you're using? So he synthesized artificial songs using a package. And if you can see this, there are 4 different notes, A, B, C, and D, and where A and B are slightly similar, and C and D are a little bit different. And the whole idea was that these A, B, C, and D can be mixed in different combinations. And what he wanted was an index which showed that ABCD would have the highest, highest kind of complexity value, as opposed to AA, BB, and so on. And so he kind of played around with this until he was happy with that. And when that happened, basically he had these discussions with us, and we said, well, but does it work with real bird songs? Does it only work with synthetic songs, or is it valid for the real bird songs? A lot of literature is there where people have described very complex songs and so on. So he went on to use his metric on various songs, songs birds that have very short songs and some that have very long songs. And he felt that this metric kind of captured this variability quite well. And coincidentally, he also found that the bird that we worked on, the Sholikula, that seemed to have very high complexity in its song. So at that point, Suyash decided to get onto field, and he color banded a lot of birds with Chiti, and he recorded them for, I think, about 3 or 4 years, 150 songs per individual per year. And he was also getting these GPS points of all the ringed birds whenever he would see a bird. So he knows the territories of all of these birds around our field site. And he used different parameters, but essentially what was happening was he extracted all the song parameters that one normally extracts, and he tried to classify notes. He used a certain kind of hierarchical cluster algorithm, And the idea was to kind of cluster the groups and to get to syntax, similar to the ABCD that he was using on his synthetic notes. Now, at this point, we also decided to kind of look at the combinations of notes a little better. And that's where he started working on these n-grams analysis, where you have the A, B, C, D, like you have these notes, but these notes can be mixed in different formats. You can either consider them as 1 gram, which is you're only considering it as single note, or 2 grams, where you have a combination of the two notes. So you have AA, AB, BC, and CD. And then you have 3 grams, which is, again, a combination of AAB, ABC, and so on. And 4 grams, where you're adding 4 of these alphabets together. So this is apparently something that linguists also do in studying various, to quantify things in different languages as well. And when Suyash looked at the repertoire size, initially when he had just the notes, which is the 1 gram, it looked like if you have about 150 notes, there seems to be some kind of saturation. And he decided to kind of collect 150 notes for every bird based on this. But then if you start looking at 2 grams and 3 grams, because of the way this adds up, it's actually, there are so many more combinations that it actually doesn't saturate at all. So how did he use the n-gram? Essentially, he had a snapshot within a particular time point. So if it's a year 2012 or something like that, he has multiple individuals. So 1, 4, 9, and 12. And you're looking at sharing of songs across individuals within the year. But then you also had individuals, the same individual across different years. So here, the idea is that you're comparing across years to look at consistency in song for an individual, but within a year, you're looking at song sharing. And we were particularly using n-grams for this analysis. So the simple kind of outcome from this was that notes, so the single notes seem to be, they are there across the years. So the same notes are not only there within, they are there in all the individuals and across the years, but it's the combination that actually was consistent across 3 years. So 2 grams and 3 grams are actually very year specific, but 1 grams are consistent across years. So this is actually very interesting. So they have the alphabets distributed across, but combinations of alphabets, you could say, that are making these signatures year specific. And of course, because we had these territories mapped out, you can see the different individuals that we were referring to earlier. Basically, what Suyash was interested was looking at was song sharing and how songs are shared as you go from neighbors and increasing distance. And very simply, what he found was that as the distance increases, the difference between songs, especially the 2 gram sharing, increases. So this was actually pretty cool, except that this is something that is well documented in literature, how songs are shared and how distance has a very important role in this. Yeah, so that's the groups that we see. And it's at this point that actually we wanted to kind of examine this with greater geographical distance, because this was all in a very small area, just a few hectares. All of these territories are put, in around a field station in a very small geographic space. So that's where Chiti came in, and although she was involved in the study, her, the canvas of at which she was examining this is much larger. And yeah, sorry, I think I didn't talk about the song consistency. Yeah. So this actually looks at consistency. It's a different representation of what I just told you about 1 gram and 2 gram. And you can see how the consistency varies, in 1 gram and 2 gram. And this is sharing across years. So this is the particular individual S4. And you can see that at 1 gram level, 59% of the notes are shared. So basically the same notes are everywhere. But if you look at combinations, which is 2 grams, so AB, BC, and so on, that's when you start finding these unique signatures where you don't see as much shared, but you see that in every year there are these specific combinations that they seem to use. It's really fascinating how that's working out. And the individuals sharing as well. So in this case, this Venn diagram is has different individuals. You can see at the 1 gram level, you can see how they are shared. You have the shared, there's greater sharing. But at the 2 gram stage, you start seeing individual signatures. Yeah, so this is actually quite interesting in how birds are doing this, how they are kind of combining these notes. So yeah, I'll let Chiti tell you a little bit about the larger geographical scale part.
So Robin just described Suyash's work, which focused on looking at Sholakili song within the same population. So we were looking at individuals and their neighbors and how songs differ as we increase in distance. So what I am looking at is our songs basically at a population level. So like we looked at songs in a particular population, which is the Kodaikanal population, which is in the extreme east. I'm looking at 7 populations across this entire landscape gradient, extending from east to west, and also 1 population, which is right down in the south. So do these isolated populations have distinct signatures in their songs? Can we identify certain dialects that are specific to populations? And another thing that we were interested in looking at is whether there is any genetic population structure across the species distribution. So we must remember that this entire landscape gradient is only about 60 kilometers from east to west. So it's a really small area. And we sampled birds from all these populations and we are looking at some genetic population structure like admixture. Next slide, please. So also I must mention that this is all data that we have collected before and analyzed. So we have more updated data that I'm currently analyzing, and we have some results and the results have been generated between our first demo session with Eduardo and right now. So I didn't want to add the new slides to mess up the order, but if we can bring it up on the screen for the purpose of discussion. But this is all the previous data. So we went and collected focal handheld recordings from these 6 populations. So as you can see, it is from east to west, and 1 population that is HIGHW, which is right down in the south. We had about 300 to 700 songs per population. Currently, we have about over 1,000 songs from every population, and we have done some analysis for that. Next, please. So we'll be focusing our results on 3 populations, which is Kodaikanal, Bandar, which are close to each other, and high babies, which is further down south. So the first thing what we did was is to do like a PCA between the spectral variables of each individually annotated note. And the spectral variables that we used were minimum frequency, maximum frequency, delta frequency, peak frequency, and song duration. And what we see is that, yes, there is no pattern. So between the spectral variables itself, there is no differentiation in songs between populations. we went a step further and then looked at the presence of these complex vocal mechanisms. Complex vocal mechanisms are nothing but the occurrences of two notes less than 2 milliseconds apart from each other, which basically means that they are overlapping notes. And what we see is that as we move from west to east, which is from grass to Kodai, the HIGHW population is down south. But as we move from west to east, we see that there is a drop in the proportion of CVMs present in a song. So basically, each dot represents the proportion of CVMs in a song. And it's significantly lower. So why is this interesting? Because we believe that CVMs are culturally transmitted. And it's not that the birds in the Kodaikanal population cannot produce CVMs. It's just that they produce CVMs at a much lower frequency. Next, please. So yeah, this just shows us the map. So as you can see, the Kodai population, which is in the east, has a lower proportion. So this actually corresponds to Robin's older data, where he did find that the grass population had a much higher proportion of CVMs as opposed to the easternmost population, which is Kodai. And as we sample across this gradient, which I have done, we see that there is a gentle loss in the proportion of CVMs in songs. Next, please. So now looking at differences in n-grams across these three populations. So how did we arrive at n-grams? So we earlier used a method known as dynamic time warping to classify the notes, but right now we are using spectrogram cross-correlation, which is basically each note, a wave file is saved and you match it with a corresponding note type. If it exceeds, if it has a cross correlation score of beyond 9, it is classified as the same note type. And if it is below a certain threshold, you classify it as a new note type. So we classified notes and we looked at an NMDS plot of the 1 grams, 2 grams, and 3 grams. So at the 1 gram stage, we see that several of the note types are shared. So that's why all of them cluster together. But at the 2 gram stage, something interesting happens. This is just I don't think the x-axis is visible, but this is just a frequency distribution of occurrence of different n-gram types. So there are certain note types that are very common in all the three populations. Now, as we go to the second ordering of these note types, which is the 2 grams or bigrams, we see that the Bandar population and Kodai population, the two populations that are closer to each other in the map, share some degree of bigrams, while the High Baby population, which is further down south, seems to have some separation and don't share the order of these bigram notes. And as we go higher up in order at the trigram stage, we also see a similar pattern. But also I must state a disclaimer out here that we need to add many more songs to this data set to get a higher resolution of trigrams. I must also mention that bigrams are nested under trigrams and unigrams or 1 grams are nested under bigrams and trigrams. Next, please. So we also tried to see whether we could depict these sharing of note types in the form of an admixture plot, where which is the usual representation of genomic data in looking at population structure. And what we see that in this case, this is just an exploratory graph. What we see is that there are note types shared across all the three populations, but the Kodaikanal population seems to have some different signatures in certain n-grams that are characteristic only to the Kodi population. So a little bit about the genomics, what we did. Here we have collected about 159 samples. Our final sampling has about 250 samples collected across the entire landscape from this is, I think, Robin, next. Oh, okay, we don't have the total, sorry, previous. From about, I think this is 9 populations out here, but we have a total of 13 populations now. And what we did was DD-RAD-seq with 2 restriction enzymes, MLUC and SPH1, which was used by Robin earlier. And we sequenced this on a Illumina NovaSeq platform. I must also mention that these results were for the de novo assembly of the ddRAD-seq data, but we also have done a new reference genome assembly using nanopore data and Illumina short-read data, and have mapped this new data to the reference genome. So the results show us that so in this case, we mapped it against the closest sister species, which is the Nilgiri Sholakili, which is found in the landscape above the geographic barrier. We did SNP filtering in VCF tools and ran admixture for different levels of K. And in this case, so in this case, with the old data, we had a lot of missing data. So this is about 75% missing data. But currently, we do have a much higher resolution of data, and we have retained about 13,000 SNPs. But in this case, we see that the high wavies population, which is in the center, seems to be quite differentiated from the populations that is Kodai and Bandar, which is above. It's not arranged in order. The high wavies population is in the center, and Kodai population shows some admixture. But in the new plot, what we see is that there is some degree of isolation by distance from grass to Kodai, and the high wavies population clusters absolutely separately. So though we also calculated FST, and the FSTs are really low, which the maximum differentiation is about 0.04 with high wavies and the populations that are in the above landmass. But if you plot a PCA of the SNPs in this in the sample populations, you see that the high wavies population remains distinct. So when we look at song dialects, we see that songs differ across populations, where at the n-gram level, or at least at the 2-gram level, we see that populations closer to each other have a higher degree of sharing than populations that are apart. And at the, this is again the old plot for genetic variation, we see that there is no population structure, but we now do see that there is population structure, with the high wavies population seemingly much distinct as compared to the birds in the Annamalai Palani landscape, which is above. So why is this interesting? What is so interesting about this landscape and the birds found in this place? We are sampling at a very fine resolution, but just within these this distance of 60 kilometers between each other, the extreme westernmost end and the extreme easternmost end receives differential rainfall. So the westernmost end receives maximum rainfall during the monsoon, which happens during May, June, July, and the easternmost end receives rainfall later on in the year during September, October, November. And though the west receives much higher rainfall in the summer months, the east receives almost comparable rainfall, but a higher peak in the east during the winter half of the month. So another thing that we are trying to do right now is to map climatic zones, which is basically classifying the entire landscape at a fine resolution with temperature and precipitation. So these birds prefer areas with colder climate and high rainfall, which is also corresponding to high elevation. So this is work done by Akshay, and we are trying to see whether climatic variation can cause barriers to gene flow. And another thing which is interesting about this landscape is that, Robin, next please. Oh, okay. So, oh yeah. Robin, should I do? Mute.
Yeah. So I think the main takeaway there is that, while we find this variation, we think that the cause of what's driving the variation is probably driven by climate, which means it's phenology. So birds in one part of the landscape are breeding at a different time than the other side. Unfortunately, there's very little phenology data So if that is the case, we should find this differential breeding time as you go across, and the very edges being very different in the breeding times. And it's a difficult bird to observe and get. It's because it skulks a lot. It's very kind of secretive, so it's difficult to get this kind of data. And one of the things that Aditya, who's now I think also at the meeting, who's doing his master's project, what he's trying to do is try and use some of these algorithms, BirdNet, which works. It doesn't work very well, but Aditya is trying to get it to work as well as it could in trying to detect when birds sing. And what you see here is the results from one recorder, this recorder, which was placed in the center of the territory of one bird, and it was recording all through the year. And you can see that there seems to be a peak in March and another in June, and there's also some activity in October, November. So this roughly matches the the time that we were Chiti was describing of rainfall, peak rainfall, and so on. But what's interesting is that there is a close match between the songs detected with the algorithm and with actual raw data that Aditya and all have been looking at. There was a AI classification challenge, BirdClef, trying to kind of get detectors working better for this landscape. So hopefully, what we will have very soon is phenology data from across the landscape just based on these detectors. So Chiti has these recorders placed out every 10 kilometers across the entire landscape. And it's been recording from, I think, May or April or May or something like that. So by next year, we should get this kind of data from across the landscape. And hopefully that'll provide us some answers to why there are these differences in songs as you go from the east to the west. And I'll end with the last bit about female song. And for that, I'll play the male song first. Yeah, seems like a really nice, melodious, long song. And then this is the female song. Now, the problem is that both males and females look alike on field. You cannot tell them apart. And we know this is male and female only after we have collected the blood samples and sexed them in the lab. So when you're actually recording them, you're probably recording them blind, or after sexing, you go back to the color banded birds and you figure out, which is male and which is female. So Rohit, is another person who interned in our group, and then he kind of figured that actually the female song seems to be quite complex as well. And in fact, he finds that the complexity of female song is probably a little bit higher than the male song. Well, we're still trying to figure that out. Basically, in terms of all the other variables that Chiti described, if you remember, note duration, song duration, frequency, in none of those, the males and females seem to be any different. They are very, very similar. And because the song seems to be a little bit short, at least this metric seems to suggest that the female song is more complex. Now, the question is, why does this have to be so complex? Why do females have such a complex song at all? And this is not very clear. We don't really know when the females sing too well, because we do know that it is somewhere around the breeding season. So what is a function of the female song? We already described that the male song is very complex. It uses all these syntax kind of things, n-grams. The stability of notes across the landscape, and they have these individual signatures within years, which are made of these combination of notes. But then what you can probably assume that there is a sexual selection role there in having more and more complex songs. But then why are female songs complex? And that's something that we are just beginning to explore. So I would say that, in summary, hopefully what we have described is that songs are really distinct across these mountains, these sky islands. And within the mountains, also there are differences in songs. They are fairly complex. It has a large repertoire of notes. I don't know if people caught on to this, but remember that we said that there are so many notes, but all the notes are shared across the landscape by all the individuals. So the only difference that you find is in how different individuals mix these combinations. So everybody has A, B, C, D, the entire range, but it's how they mix them. Or some individuals do A, B, the combinations. That's what is unique. And those combinations change across years. Now, while I say this, you wonder how birds recognize each other. And I'm 100% sure that they are not using a complex algorithm like the one that we are describing. Because if like Chiti does these playback experiments, you hit play and birds immediately know if it's, they know each other very well. So there is something additional that is there, which all our complex analysis is still not capturing. And yeah, Chiti also showed that the songs are different, not genetics, but there is some genetic differentiation, which seems to be turning up more recently. And female song is still a black box because we need to understand what time the females sing, when do they sing, what do they sing for, the context of the song. I think it can probably only come from a lot of field observations. So we are slowly beginning to couple video and other additional observations at nests for all of these individuals to try and tease this apart. Ultimately, we're just completely fascinated by how complex these songs are and how the sharing and these mechanisms work in this landscape. So yeah, we have a whole bunch of people to thank for permits and funding and so on. And with that, I'll end and take questions. Chiti, did you want to add anything else that I missed out?
Okay.
Yeah, thanks, Eduardo. I hope we are roughly on time.
Perfect. I think this is fantastic. It's really what we think it benefits us all, because we may get some questions from some of our colleagues who may need to go at noon, and then we'll have more time. So thank you, Robin. Thank you, TT, for a very interesting presentation that I'm sure is going to generate lots of questions and conversation. So once again, if this is your first time participating in the FINE seminar, what we normally do is that we ask that you type in the chatting just a question mark if you have a question you want to pose. If you're a student, please type the question mark and an S. A student or postdoc, we really work hard on giving priority to our younger colleagues. We want to build that kind of community. And so we want you to feel comfortable asking your questions. And I'll be taking the questions in the order that they are posed. If you need to go, I'll take one more time to remind those of you who are going to be leaving at noon that next week, let me see if I have it handy. I can open it right away to next week. One more time, let me show you, because we're going to have another very exciting presentation. Just very quickly, I'm going to stop. So next week, we're going to hear from Jen Moss from Virginia Tech University in the US, discussing unpacking thermal effects on social behavior through integrative approaches. So that's next week. I also want to invite you all. Most of the times we open the Zoom a quarter, 15 minutes before the talk, in case you want to stop by and say hi and meet the speaker more personally. We can do that before we actually start the talk. So with that, I think we're ready to ask questions to our colleagues today who are so kind to be doing this 8, 9, or 10 p.m. in the evening. So do we have any questions already in the chatting? Go ahead, type a question mark if you have them. If not, I'll jump on to asking some of the questions that I have myself. I see some new participants. Any questions from the audience? Okay, I have one myself, which is not really a question, but maybe it'll get us. Robin, thanks for saying that. One of the things you were hoping is to really brainstorm together about some of the findings you're reporting and where to go. Can you, for somebody who is not, I mean, I'm not really that familiar with the literature on perception, on sensory ecology. Where are we, at least for the study of birds? I'm quite familiar, and we hear a lot of talks and research being done on the production of songs and the production of vocal stimuli. Much less, because of course it's much more difficult, on the perception of them. And so it's like, where are we in your project, if you're doing any of that, or more general? I would really benefit from hearing from you guys who are more familiar with the topics. What are we doing? What are we seeing the field do to understand how all these signals are being perceived? Because without knowing that, we'll never have the full story, right? But of course, we know how difficult it is.
Yeah. Chiti, do you want to?
Signal perception in the sense of response via playback experiments, that is quite common.
No, I'm thinking even more. That's what I'm saying. This is very challenging. I'm thinking black box. For most wild taxa we study, it is still a very big challenge getting really to understand how the brain is processing all these signals. I mean, selection is not going to, I mean, the genes that you'll be looking for these things are going to be acting at a much more mechanistic level than where the bird responds to a playback or not, right? That is not what's getting selected. So that's what I mean. Are these taxa that people have brought into the lab to start understanding those things? And I'm very aware of how challenging it is, but you guys I'm sure you know more than I do. What progress have we done in the last 3 decades on this topic? I mean, I'm thinking of Michael Ryan's work with the frogs and but those were frogs, how he was able to really look at both ends of the communication process in a very mechanistic way, almost neural way.
I think there's a lot of research being done on signal communication in at least the zebra finch, which is the bird model organism, and they do look at a lot of responses and stick electrodes at different points in the brain to see. But for birds like the Sholakili, which is wild, A, we don't have permits to put them in a captive population, and B, they probably won't survive in captivity. So, and also, I think defining complexity is very subjective. So, zebrafinch researchers might define complexity in one way, and we might say it's not that complex. But in our sense, complexity is defined as probably differences in note types, and our note types are also with different, let's say, peak frequency contours, but which is probably not as important in zebrafinch, which have like stacks of vocal representations, which is defined as more complex.
And
So,
I apologize if I missed it. Have you done playback experiments? How likely is that? Given, I imagine this must be a very difficult habitat to work
yes.
on. And right, so that's
Yes, yeah, that's absolutely correct. And in fact, it was one of my chapters which I had to unfortunately abandon because it's so difficult to get a response for a bird that is an understory bird in a tropical environment, in a cloud forest habitat. So you see the bird, and then the playback starts, and then the bird is gone. So you can't track the detection distances as well. where it's moved, where it's gone, how high it's gone, how low it is. So, but they do respond seemingly to songs from different populations. But what is the extent of response is something that we are still trying to measure. So we are going to give it another attempt and see how that works. But it is quite difficult in the wild.
Before I take on the next questions, just to get a bit of more sense of the natural history, over how big is the whole territories, or on a given week, an individual, how much will that individual move? How big of an area?
I think it's so Yash, if you're here, do you have a number to that? But it's a very small area.
Yeah, it's under a hectare, I would say.
Yeah.
Okay.
Yeah, much lesser than that also.
Okay.
But the best thing we would have liked to do is to put GPS trackers on them, but
Yeah.
that's not been possible. That would have been the ideal way to map territories. But we have followed individuals because they're color banded individuals. So we have followed them across several years, and we have locations, and we have just drawn minimum convex polygons to map our territories every summer.
Good. Thank you. We have a question from Heather Kenny, and your name is Heather Kenny Dadela. Heather, please. If you can please, I forgot, if you can please introduce yourself, where you're joining us from. People join from all over the world, and what's your institution and what do you do?
Great, yeah, thank you. Yeah, I'm Heather Kenny Dudella. I'm a PhD student at the University of Colorado in the United States. And but my husband is actually, his family is from Tirupati, and so I have visited. And so next time that we're there, I would love to meet up and connect with you in person if that's of interest. Yeah, so I'm working on sexual selection of plumage color in barn swallows in Colorado right now for my PhD. But I really enjoyed your talk, and I was curious sort of if you know what is the lifespan of these birds, and if they so they maintain a territory, and for an individual, like, is that territory, do they maintain it all year long or like across years, or do they move? And is it like a breeding pair that has a territory, or it's individuals that have territories?
Yeah, I think, Robin, do you want to? I'm just quite excited to take
Yeah,
it.
yeah. I mean, yeah, you have to hold Chitti back sometimes. Yeah. So, so when Suyash started doing his work, trying to, look at these banded birds, that's when we found that a lot of individuals are fairly stable, and so the territories kind of stay. There's one individual, yellow, which it was our favorite in some sense, because it was just around our field station, and it basically once it disappears, it's gone. Like, you don't find it anymore. So you don't find these territories changing as much. We've followed now, at least I think for the yellows, 2 generations of yellows.
Yeah.
It's young, and then that one had another young, and we knew where they went. So I think they're fairly stable, but the way territories form and the new territories form are really interesting, because you see one bird with a very, where the song is not totally crystallized, and it comes in, and then slowly that develops a new territory. So although the core territory stays, there are new individuals that come, and there's small shifts that happen. But essentially, the core still remains pretty stable, at least through 4-5 years that we've observed them.
And to add to how long they live for, Robin has been doing mist netting about 10 to 15 years ago in the landscape, and we went a few years back, and we have been doing mist netting, and we have actually caught Robin's old birds, which are about 12 to 13 years old. So this bird is like a really tiny bird. It weighs about 20 grams, and it clearly lives for a really long time. We don't know till when, but it's definitely 12, 13 plus.
Wow, that's really cool. I guess just one follow-up about the territories. Do you know if there are aspects of the vegetation or anything that's really correlated with territory quality? And is like territory quality at all correlated with any of these song aspects?
Yeah, Chitti, I think that was Chitti's chapter that she
Another,
dropped. Yeah,
yeah,
so,
it's like several chapters
Well,
that I've had to
it's
drop.
very
But
interesting question.
yeah, it's the gold that we were after, but it's very difficult to collect that kind of data, especially to kind of, to find the differences. You have to be able to quantify quality in a way that I mean, we're just beginning to scratch the surface of the system. So I think we are not yet ready to ask that question. At some point later, when we have maybe a lot of these nests monitored, we have maybe nest cams and a few more individuals and some generations, I think we may be able to get to that. But
Well,
thank you.
thank you.
Nice question. Thank you.
Thank you, Heather. The next question is from Snara or Snada. I don't know how you pronounce it.
Snada.
I
Yeah,
I
thanks.
haven't forgotten about your book. Trust me. Bear with me.
No, I know. Please take your time. You're really busy, but I'm
But
still
please,
really
again,
excited. I'm
introduce
getting that
yourself
book.
and remind me where
Yep.
you are. Yeah.
Yes. So I am a PhD student at the University of Portsmouth, that's in the United Kingdom. And I'm studying anti-predator strategies in colonial spiders. So these are the tent web spiders. We study them in Spain, and I'm hoping to include some field sites in India because I've seen them in my backyard back in India. First of all, please let me thank you for this amazing study. I've also very interested in birds, and like acoustic communication aspect is one of the most obvious. So the n-gram analysis it's like just blew my mind. It's so good. I have a question about I will actually follow up from Heather. So do you know how stable the breeding pairs are between breeding seasons? And have you identified any kind of signature of duets in these birds, in the Sholikolas?
Uh, the short answer is no and no, in the sense that we don't think that they'd see. Yeah, so once the short answer is out of the way, I can actually explain a little bit more. The thing is that we just see. I've been working on this bird for 20 odd years, and we knew that there is probably some female song, but if you go by the literature, the female song is supposed to be a small subset of the male song. And what we find is that that's not really the case. In fact, the female song is just about as complex. And we found this just 1 year back. We're just beginning to uncover this. I don't think we have seen anything like duets. We do see a lot of song matching bouts that happen between neighboring males. That's very common, but not duets in the sense that the males and females are singing in some coordinated fashion. So that's not yet documented. It would have been interesting if something like that was there, and it could probably explain some of the complexity, but no, we don't. We don't have that. Yeah.
Yeah, I understand the difficulties of working with them. I have been to just once, and just that image in my mind, it's like you're doing a great job finding all these complexities. I'll just add one more question to this. So I was thinking, do you feel like the female song's complexity, could it also, just theoretically, of course, could it signal some sort of communication to an extra pair mate, potential sort of extra parentage or something going on? And yeah, do you plan to follow on a parentage analysis, Chiti particularly? Or would you be interested in that?
Yeah. So as of now, see, the pairs that we followed have been stable, but there have been very few. We've only followed them over the last 4 years or so. But I suppose there could be some. I mean, I think most passerine birds, there's going to be some extra pair paternity. But the role of the female soliciting that with the complex song is a fairly complex idea by itself. Uh, yeah, I mean, yeah,
Yeah.
I don't know of systems where both are happening in that manner, where males have such a elaborate complex song, and then so do males. I mean, so do females. Um, I'm curious to hear from others if they have ideas on that. But
Yeah, I'll just add, this is coming from a very old paper because I was doing a short project with Professor Wolfgang Goenman during my master's. And this is called the Coyness model by Wolfgang Wickler. Yeah, it sort of explains why that like more time investment and learning to duet could actually reduce the chances of extra pair paternity. And yeah, I was doing a meta-analysis, so we didn't find it very common in the most of the passerines we looked at. But yeah, I don't know what you would find with Sholakili. It's so interesting. Good luck.
yeah,
And
thank
thanks for the
you.
answers.
Yeah, thank you so much. Yeah.
Before we go on to the next question, Lauren, if you can spare me a minute. Sana, you were making reference to Wickler's paper on monogamy? Did you
Yes,
say?
yeah. One of
And
the
so
two, like,
tell me, so this was part of your master's thesis where you were looking at, there's a number of primate taxa where the male and the female produce duets. It so
yes.
happens that in most cases, those are pair living taxa. So can you tell me again, what is that? We can follow up later, but what is that you were looking or you're interested in?
Well, we were looking at the passerine birds. So it was a meta-analysis with the question that do duetting pairs have less extra pair youngs in their clutches? And yeah, it was mainly focused on passerine birds. But I know which you were talking about, and that's exactly how I took up. Like I read about that, and then I thought that this could be a nice rotational project. It was not my master thesis though. And yeah, let's definitely follow up on this. It's very interesting, Wolfgang Wickler's paper. Yeah.
So it's very difficult to study duetting also in primates, and even more difficult for any association we may find between duetting and extra pair paternity, making the causal connection is going to be yet extremely complicated. There's so many things that could be associated with duetting that are more important. Anyway, thanks for letting me know. Yeah, hey, Lauren, you're next.
Yeah, Clay.
Thank you, and thanks for this really interesting talk. And I really appreciate you giving a seminar so late at night. It's much appreciated. For my question, I just wanted to say that people on YouTube, if you have a question, post it in the chat, and I'll put another question in the chat here. I have a number of questions, and I'm completely naive to this kind of work, so please bear with me. I'm thinking about the data you showed in the male versus females. There was a figure where you showed NVI, which I think is note variability index. And my first question is, how big of a deal is it that like, what's the scale of variability on there? So are we talking like, reference to grams versus kilograms scale? Or are we talking, 1 unit is not much variation? And the reason I asked that is because the variability was greater for the males than the females. And I'm wondering if that's enough variation to be interesting to look into to see, like, why are males more variable than females?
No, I agree with you. I think that one of the problems is that this is still very preliminary data, the female data. And so we are still trying to figure this out, but it's just at the moment, the error bars are really large. So it looks like there are females which have high note variability index, and there are some males with lower note variability index. Males, we have a lot of data, but it's with the females that we don't have as much data. But I think that these are not very large differences. Rohit, I can see you're here. You want to comment? You have to unmute yourself.
Yeah, hi. Can you hear me?
Yes.
Yeah. So the note variability index that we did comparing males and females, because the data is very low for the females, and so we've taken because the data is very low for females, I think it shows. That's the reason why it shows that. But I think if you have a more larger set of data, then we'll have a better idea of how exactly the note variability index plays a role in comparing both the males and females.
Yeah, but I think the main takeaway from that figure was not that we were not actually trying to say that females have greater note variability, but it's just that it's comparable. And that, I think, was a bigger kind of surprise for us, because we expected the songs to be much simpler, maybe a few whistles and, small, if all the literature points to that, except, there are researchers who focus extensively only on the female song. And they've constantly been saying that tropical birds have a lot of female song, tropical passerines, and it's all been undocumented. So we are very kind of excited that, we've kind of stumbled on the system which also has female song. Just adds a layer of complexity to it.
Cool. Thank you.
Yeah, thanks.
Edward, are you muted?
Yes, Rag, I think you
Thanks.
come up next.
Yes. Yeah. Thank you, Robin and Shitty. I enjoyed your talks very much. I have read all your papers, but all of this was still new to me because, as you said, for the others, I am Raghavendra Gadakar from the Indian Institute of Science, Bangalore. And I have known Robin for a very long time and absolutely delighted that he was chosen to speak in our fine seminar series, and he and Chitty did such a wonderful job. I would like to persuade you, both of you, to say a little bit more about how your work, or how you would like to your work to fit into the bigger picture of behavioral ecology, evolutionary biology, speciation, conservation biology. Why are you doing these things? How would this fit in the bigger picture?
Yeah. Yeah, that's in some sense, what we are trying to do with these birds in the Indian system is not very common. You don't have marked individuals. You don't, like, individual based studies are very little in birds. There are just a few studies, maybe 4 or 5 that happen in India. So having this kind of data is very powerful. This is despite the fact that probably birds are the most studied taxa in India. Most, like, across biodiversity, if you see where is data, most of the data is on birds.
Sure.
You'll find that much of it is observational data, which is, I mean, ours is also observational in some sense, but in the sense that it doesn't involve marked individuals. And that provides a very different perspective to, the kind of behavioral ecology questions that we are asking.
Mm-hmm.
So I would think that this is, when we talk about birds that have a great diversity of songs, just the idea of recognizing cultural diversity as a part of this whole sphere of, let's say, biological diversity and genetic diversity. I think this is something that I am trying to convince, let's say, conservation managers about. And certainly, even in terms of, we live in a world where everything is viewed with a lens of how applied it is. And I think that there's value to just understanding how birds behave, and there's a beauty and joy in just doing that. But I think even in terms of understanding how these populations that are isolated, and you can tell them apart by their song, can be a very strong kind of message for conservation biologists. Because what you're trying to say is that we spend, like, thousands of dollars trying to sequence these genomes and trying to look at the differences in populations. And the differences are recent, so you don't find so much, so many differences. But then the cultural traits vary rapidly. And you are able to use that as, to be able to understand, let's say, impacts of deforestation. It's isolation ultimately that this reflects. So which is why the Sky Island system is a good system to kind of understand this bigger picture of how isolation and fragmentation, what are the kind of impacts that are there, and how can you measure it? Yes, you can measure it with genetics, but that is a different tool. I think that the fact that we're able to use bird songs to show some of the same kinds of effects that, let's say, tiger scientists, biologists, or other, conservation geneticists show. I think that's an interesting idea.
Sure, thanks.
Do we have any other questions from the audience? If not, I would like to ask our speakers something else. So, and I don't know whether your invitation to think together about possibilities related to this. When you were describing that you lack as much data as you'd like to have on phenology to assess the seasonal changes, you really, I mean, maybe I got confused, but you're talking phenology in terms of assessing plants, because then you were describing automated assessment of phenology. So if you're talking about plants and you feel like we know how time consuming it is, I wonder if you could go back, getting samples of trees to look at tree growth as a proxy for change in the, I mean, yearly, and look at tree rings. If somebody could sample those to see if there's any proxy of what the weather may have been like in the last 10, 20 years, whatever time scale you're interested in. But I wasn't sure what you meant by automated phenology data. I think you're referring to using the birds as a proxy for food. But
yeah. I can explain that. Maybe I just went through that a little bit quickly. Essentially, what's happening is you have this east-west gradient on this mountain. And what Chiti finds is that the songs are changing, slowly as you go across this landscape, until one point where there is a slightly more rapid change. So that place where this turnover is happening, actually, what we think is it's a change in the climatic pattern. So how does a climatic pattern affect a bird's cultural trait? And that is where we think that it's because they sing at different times. So if you're singing at one time when closer to the rains in one season in one area, and you're singing at a different time because the rains and the resources are at a different time. So that's the phenology mismatch that we're talking about. So the East and the West get rains at different times of the year, and because of which the birds are singing at different times, and hence there's like an isolation that's created, and there's this cultural isolation that's happening just because of how climate is driving this pattern. So ideally, what you would want to see are those radar plots of when birds sing all across the landscape. And you should see that exactly when that line that Chiti showed, where the rainfall pattern suddenly changes. That's where you should see that change in phenology. You should be able to see that birds actually sing at a different time from there onwards. That's our hypothesis.
Right.
So,
No, I got you. Yeah, okay. Yeah. Anytime we've been doing phenology for 28 years. I never thought I would be looking at trees for such a long time. Let me say that I wasn't a fan. I'm not a fan, and my colleague who's been doing it takes a lot of time. And what's been fascinating, this is a manuscript we have in revision. We have done now very detailed analysis of the very limited data we have. Those are two. I mean, you can analyze all you want, but if that's right, and trying to understand if phenology, availability of food in the forest, is in any way related to the likelihood of a pair of almacles having an infant in any given single year. And we're not finding any pattern that we find convincing. And right. Now, am I saying that food has no bearing on infant production? No, but most likely my guess, my hypothesis, that we're just not getting at the specifics, at the nuances of how you measure food availability. Right. You just mentioned hypothesis in a way that really, really is assessing what the animals have and what the animals take in to influence, in our case, production. So it's, this is because every single month we need to, I'm saying, okay, is it worth, should we keep doing it, and what for? Because we cannot do it unless we do it very kind of coarse grained. We don't really have the kind of data we'd like to have what the animals are actually eating.
Yeah, I think you've hit the nail on the head. Ideally, this is what we should be doing. We should be because these are insectivorous birds. We should also be looking at the exact resources that they are tracking. But because we can't do that, we're just using a proxy.
Right.
And we're not even actually observing the birds. We're just putting these recorders out there, automated recorders, and getting a proxy of that as well. But yeah, I mean, there's only so
As long
much.
as you're, I mean, like I always tell students, provided you're explicit about it, that you know it, right? At the end of the day, we just want to know what we know. Yeah, thank you.
Yeah. Okay.
Lauren, you have another question?
I think so. So to follow up on what you were just talking about, and I apologize if I'm off. My brain was thinking of the other question. Can you use satellite data to track greenness, which would be an index of some of this ecological resources you're talking about?
Yeah. Unfortunately, I mean, yes, we tried looking at various satellite data also. That one of the students actually just did entirely that. The problem is that it's not that the extent of rainfall is different. It's just that it just comes in at a different time. So the habitats are actually very similar. So I think what really is changing is like what Eduardo said, it's probably the insect abundance. It's the peak of insect abundance activity that is shifting. And that is what is probably actually driving the bird breeding period. But that's just so difficult to track, across a landscape. So yeah, I wish we could find something that that worked at a larger scale.
Can you collect guano? Is there a way to kind of determine what they're feeding on through those kinds of samples?
Chiti, you want to say something?
Yeah, we thought about it, and then at the last stage of mist netting, we started collecting the poop samples. So it's all sitting there in the fridge. We've got to do some meta barcoding for it.
Cool.
So yeah, that'll be something interesting to look at. And also something that we missed saying is that this landscape has a lot of vegetation modification as well. So a lot of the grassland area has been taken over by timber plantations. And these timber plantations also support these endemic birds. So that's another interesting angle. So the birds are present in a different habitat, but what are they feeding on, or what is driving their persistence in terms of resources is also something that is yet to be looked at.
What are the differences in noise levels in these habitats?
It's not human, if it's, I mean, like anthropogenic noise, not much. They're all protected areas.
Okay.
More or less.
And then, can I ask another one, Eduardo?
Yeah, go ahead. We don't think. Yeah, go ahead.
I think this comes back to Raghavendra's comment about the broader context. I'm thinking behavior here. How would movement of animals by dispersal or through exploration, I mean, how different are these dialects? Are they settling in different areas, and then they would bring that sort of component of their song with them that then would be learned by the animals near them? Is there any of that going on, or is it so different between these areas? I'm looking at the figure with the orange, red, and black color in it with the territories. I think I remember you said that there was difference in the dialects in those distinct regions.
Yeah.
Chiti, how is that established, I guess, is what I'm getting at.
You can go first, but I want to add to it also. Yeah.
I think the map that you're looking at is, I think, for one population. So it's different territories in different territories of birds within a single population. And as distance increases, we see that the song sharing also decreases. But between populations, we only did 3 populations in the graph that's shown in the presentation. But I have about 7 populations now, and I'm still running the analysis. It's hanging. But hopefully very soon we'll have some results to see the gradient.
Yeah, but I think overall, like the big picture is really fascinating that all of these birds have the same set of,
No
alphabets.
types.
Yeah, the same note types, and that's getting shared all across. So definitely there is movement across, and that's reflected in Chiti's analysis. And that is, the data that she has is from whole genome kind of sequencing data. So it's a lot of SNPs that's gone in there. I don't think that, we don't need to add more data. There's no more resolution that you need, at least with the current technology, to show that there is movement. So there is movement both, and connectivity in terms of gene flow as well as these cultural traits. But it's just that the changes in the cultural traits happen rapidly, as you saw in that example of one population, right? You can see that individuals have different signatures every year, despite using the same node, and neighbors have different signatures. So it's just like, there is this isolation that happens with scale as you scale further and further away. Not The differences are not because of lack of gene flow, at least at this scale. Is that?
May I ask one more question?
Sure, sure. Please.
Are there age-related differences in song signatures?
There are some, at least the early songs, the first few years, I was explaining, I think, to Heather that as the birds settle in a territory, you find a lot of garbled songs and things like that. And you even wonder if it's the same species. And then, like the next year, it's actually singing beautiful songs. And so the sub-adults definitely have a very strange kind of song. And by the second year, the birds have, a full repertoire, and it's sharing songs with neighbors. You can actually see those songs being shared with the neighbors. So new individual comes right in between a couple of territories, and then it's kind of picking up the songs from the neighbors. So definitely there's an age effect. What we don't know is through the lifetime of the bird, like maybe 5-6 years, how the song changes, if the complexity kind of goes down. That much we don't know as yet.
Thank you.
Yeah.
Heather, delighted that you have more questions. Yeah, keep going.
Yeah, I was just curious if with your genetic data, if you were able to look at kinship, or if you have any like pedigree data to where you can like disentangle sort of the spatial relationships with the actual familial relationships, and how that is related to like song similarity or differences.
Yeah, so the short answer again is no, we didn't have that, because I mean, there seem to be two ways of doing this, where we could do a population level relatedness, but for the actual kinship, the problem is that you need a lot more observational data to at least assign paternity to one of the parent. And that seemed to be where we got stuck. So we have the blood samples, but we just didn't have enough data to assign one parent. If we knew that, that would have made things better. So like I was saying earlier, that we plan to have these nest camps. And I think when that happens, at least we'll know the mother clearly. I mean, there's a lot of it's a little muddled right now.
Yeah, I just wanted to mention, so we're doing kinship analysis with the barn swallows, and we have low coverage whole genome data. So it would be, I think we use like 90,000 SNPs, so like more SNPs. But we did find a program where you didn't have to give any prior information about the parental identities. And so I can share that with you. I'm not sure if it would work for the data that you have, but
That would be excellent. Yeah, thank
yeah,
you.
I definitely appreciate the struggle of finding like what can I do with this data.
Yeah, thank you so much. Yeah.
I don't see any other questions. We're a small group, I think, and it's late for our speakers. I'm going to start by definitely finishing, if you learn and agree, our YouTube streaming. So we thank our community joining us via YouTube. Remember that accessing the Zoom meeting, there are no passcodes. Everyone is welcome. So please, if you would like to join us via Zoom, you're more than welcome to do that. So with that, I will stop the live stream, and to our YouTube community,